So I welcome you all for this today's lecture. Myself, Dr. M.M.Chopade from Mumbai Veterinary College, Mumbai, and I have been assigned the topic of multiple alleles and sex-linked inheritance. So today we will discuss the problems related to the multiple alleles and sex-linked inheritance. So directly before going directly to the examples, we will discuss about the multiple alleles. Now, in a normal case, we know that all the characters of the individuals are governed by the genes, and each gene has a two copy of allele. One copy is received from the father, while other copy is received from the mother. But in case of multiple alleles, this is some of the different concept. Here, there are many alleles at a single locus. So when there are three or more kind of allele occupy the same locus in a chromosome, then we call it as multiple allele. Now, how these multiple alleles are formed? Actually, these multiple alleles are formed from the wild allele due to the mutation. So the number of times the mutation takes place in the wild allele, that much number of copies of the multiple alleles get formed, and they occupy the same locus at the chromosomes. Now, these pairs of allele functions to produce the different effect of the trait and interact independently. Now, to have more understanding about these multiple alleles, we must know the properties of these multiple alleles. The first thing is that these multiple alleles occupy the same locus on the chromosome, and they are also responsible for same character. Now, as these alleles occupy the same locus on the chromosomes, there are no any chances of crossing over between these alleles. So there will be no any crossing over occur in between these alleles. And the wild form of allele is always remains dominant, because all other forms, that is mutant form or mutant copies of these alleles, they are formed from the wild allele. So by default, the wild allele will always remain dominant over all these alleles. Then next is there is a dominant and recessive relationship between these alleles. As I told you that the wild allele is always dominant in this series. So there is always dominant recessive relationship between these alleles. And when the wild type of allele is crossed with any type of the mutant form of allele, the phenotype is always wild type. Okay. That means these are the same properties. The wild allele always remains dominant. Now we will see some examples on these multiple alleles. The blood groups in human being and the coat color in the rabbit are the best example of this multiple alleles. Okay. So as far as this coat color in rabbit is concerned, there are four types of coat color in case of rabbit. First is the full color. Full color gene, actually this full color allele is the wild type of allele, or we can say that it is the original allele which is responsible for the coat color. And from this full color, the chinchilla, Himalaya and albino coat colors are formed by the mutation. So here the capital C indicates the gene for the full color. So the genotype of full color will be capital C, capital C. But and the genes for the chinchilla is CCH, Himalaya is CH, and albino is small c. But here, as we know that this full color is dominant over the chinchilla, so there will be four genotypes for the full color. First is capital C, capital C. Second will be the capital C, capital C CH, capital C H, and capital C small c. In the same way, chinchilla will have three color because three genotypes, because it is next to the full color. Then at the third place there will be Himalaya and albino. So the hierarchy of dominance will be, first will be the full color. Full color will be dominant over all these three coats color, followed by the chinchilla, Himalaya and albino. So there will be four genotypes for full color, three will be for chinchilla, two will be for Himalaya, and one will be for albino. Okay. The second example for this multiple allele is the blood group in human being. We know that in human being there are 4 blood groups: A, B, AB, and O. So what these blood groups are actually? These blood groups are discovered by the scientist Landsteiner in 1900. He found that there are some antigens on the surface of RBC of human being. So there are some human being whose RBC containing the antigen A. So they classified this individual as a blood group of A. Similarly, some of the individuals having B the antigen on their RBC surface, so they are classified into the blood group B. And he found that there are some individuals which don't have any antigen at their RBC surface, so he classified them in a blood group O. But then in 1902, the two of the students of this Landsteiner, they discovered the 4th blood group, that is AB, which is a very rare blood group. And his students found that there are some individuals which carry both the antigens at the surface of the RBC, and then these individuals will be identified with the blood group AB. So in this way, there are 4 blood groups in case of human being: A, B, AB, and O. Now the individual which are having A blood group, they are having anti-B antibodies on their in the serum. The reason is that if that individual who is having A blood group will receive the blood from the individual with B blood group, then at that time this anti-B antibody will react with that antigen B and it will be destroyed. Okay, so that for the protection of that A blood group, the anti-B antibodies are there in the serum of that individual. The same is the case with the B individual. The individual which is having B blood group, they have anti-A antibodies in their serum. But this will happen, these antibodies will protect the individual from the clumping for only first time. But if the same individual will get the blood group from the other, other blood in the second time, then that time there will be clumping and there may be the chances that that individual may die. So in this way, these are the 4 blood groups: A, B, AB, and O. So the individual who is having AB blood group, they will not have any antibodies. Similarly, the individual with O blood group, they will have both the antibodies A and B. And therefore, the individual with AB, he can receive the blood from any of the individual, because those individuals, they don't have any antibodies in their serum. While O blood group is a blood group which cannot receive the blood from any other blood group, but he can donate the blood to the other individuals, any of the individuals. Okay, so therefore O blood group can be given his blood to any of the individual. Now here these are the genes which are we denote this A blood group with IA gene, B blood group with IB gene, and O blood group with small I gene. Okay, so the genotype for A blood group will be IA, genotype for B blood group will be IB, for AB it will be IA, and for O it will be small I. Now, these are some of the possible blood groups of the offspring based on the blood groups of the parent. Now, most of the time, the blood group of the parents, they can be used for the parentage testing. That means here you can see in the first column, the blood groups of the parent has been given. And in the second column, the blood groups which may be occur in the children has been given, and in the third column, the blood group which do not occur in the children is given. That means the individual, if the father and mother both are having O blood group, definitely the children will having O blood group only. There will be no other blood group for that particular child. Similarly, if the parents are of O and A blood group, the child will also be having either O blood group or A blood group. that child will not having any other blood groups like B or A. So in that way, this chart has been given, and this chart can be used for the parentage testing also. So the first problem we will discuss that in a rabbit with chinchilla coat, when it is crossed with a rabbit with full color coat, would it be possible for both albino and Himalaya coats to appear in the offspring? Now here we know that the parents, they are having chinchilla coat color and full coat color, and the question is that whether it is possibility that the phenotype albino and Himalaya will appear in the offspring. Now, whatever the phenotypes of the offspring we are discussing here, they are not means parent are not of any of these phenotype, that is albino or Himalaya, because it is clearly given that parents are having full coat color and chinchilla coat color. Now we have to test whether there is any possibility of the children to have albino or Himalaya blood group. Now here first we will see the possible genotypes for the full color. So full color, as we have discussed, the genotypes may be either capital C or capital C CCH, capital C CCH, and capital C small c. Similarly, for chinchilla, these three genotypes are possible. So if we want albino or Himalaya type of offspring, then it is a known fact that both of the parents should carry, or either one of the parents should carry the gene for the albino or Himalaya, isn't it? So this first genotype, that is capital C and capital C CH, will not produce any albino or Himalaya type. So either capital C CH or capital C small c, these are the two genotypes which can produce the individuals with albino or Himalaya. Similarly, in case of chinchilla, capital C CH and small c will be responsible for production of albino type of offspring. Okay, and this definitely from this capital C CH, capital C H also we can get the Himalaya type of offspring. Now these are while seeing the genotypes itself, we have some of the idea regarding this progeny of these parents. So these are all logical questions. So here in the solution, we can see that if we cross the capital C with that is pure this full color rabbit with pure chinchilla, we will not get any Himalaya or albino in the offspring, as shown in this first case. Okay, but if you see this second case, here it is capital C small c, then crossed with capital C CH small c, then here the gametes will be capital C small c from this capital C small c, and from the another parent the gametes will be capital C CH and small c. Okay, so by crossing this we will get the albino type of individual as a offspring. So here the possibility of getting the albino is there when there will be heterozygous condition in case of parent, as far as the genotypes are concerned. Similarly, here if we want the Himalaya type of progeny, then here we have taken the genotype capital C CH, when crossed with capital C CH with and small c, we can get this Himalaya progeny. Sorry. So these are our logical examples. Okay. Then second example we will see, that is in following problems on blood groups, determine the genotypes of parents. Now one parent group A and other parent is of blood group B, but all the four groups are represented among the children. Now here we want all the four groups among the children, and one parent is of having blood group capital A sorry, one parent is having blood group A and other parent is having blood group B. But we want all the 4 types of individual as a offspring. Then definitely for getting O, either of these parents should carry the gene for O blood group. Therefore, their genotypes might be definitely for A blood group, it will be IA small I, and for other it will be IB small I. Unless and until these genotypes are there in the parent, we won't get the this O blood group in the child. So that is clear. Then in the second, both parents are of group A, but 3/4 of the children belong to the blood group A and 1/4 to the blood group O. Now here we want 3/4 of the offspring of blood group A. So for that, we must have the first parent as IAIA and second will be of IA small I. Okay. Then only we can get this. Sorry, first parent should be having IAI and second will should also have IAI. Then only we can get the O blood group in the children. So I have given the solutions for this. For the first example, we have seen that one parent is having blood group A while other parent is having blood group B, but all the 4 phenotypes should be there in the parent. For that, here I have crossed this IAI with IBI. Then the IAI will produce the gamete IA and small I type, and IBI will produce the gametes IB and small I. So from this, when we cross it, we will get IAIB, IAI, similarly IBI and small I, small I. So here we will get all the 4 types of phenotype in the offspring. Now in the second case, as I discussed that, we want both the parents are of blood group A, but 3/4 of the children belong to blood group A and 1/4 of blood group O. So here the genotypes of the parents will be IAI, IAI. So from this we will get IAIA, IAI, IAI and II. So here 3 of the individuals will having this IA genotype in their IA gene in their genotype, while one is individual will having both small I. So in this way, both there will be 3/4 of the parent belonging to blood group A and 1/4 belonging to blood group O. The next condition is one parent is AB and other is B, but the children 1/4 are A and 1/4 are AB and half of are B. So for this situation, we have to be the genotype of parent IA, IB and IBI. So when IAIB is crossed with IBI, we will get this IAIB as AB genotype, IAI that individual will have A genotype, A blood group, A phenotype of A blood group. Then IBIB, which will have B blood group, and last will be IBI, which is having B blood group. So here, as per the conditions, here the children 1/4 will be having A blood group, 1/4 will having AB blood group, and half will be having B blood group. So same is shown here. This 50% will having the B blood group, and among these 2, one is having AB blood group and one is having A blood group. So they will be 25-25%. So in this way, you have to when you read the example itself, you should have understanding about the genotypes of the parents. Okay. Then third example is, if a father of blood group A and mother is of blood group B have a child of blood group O, what groups are possible in their subsequent generation children if the individual with blood group O is mated to all other cousins? Now here the genotype, the phenotype of the parent is A and B. Okay, but the phenotype of that child will be O. So in fact, the one of the parent will have genotype IA, small I, while other parent will have IB, small I. Then the child will be of blood group O. But there will be not a single child only. The next question is based on this first condition only. What groups are possible in their subsequent generation? That means F2 generation. If the individuals with blood group is mated to all of his cousins. So we will see the solution for this example. Here, suppose the father is having the blood group A and his genotype will be IAIO, while mother is having blood group B, she is having genotype IBIO. Now, either you take IO or you take small I. These both are same things. Okay. So here IAIO indicates the genotype of the father and IBIO indicates the genotype of the mother. Now from here, in offspring, the genotypes will be IAIB, IAIO, IBIO, and IOIO. Now this child with IOIO genotype is having O blood group. Now our next question is based on this. When this child with O blood group, that is IOIO, when it is mated to rest of its cousin, means IAIB, IAIO, and IBIO, then what will be the possible phenotypes in the subsequent generation? So when we cross this IOIO with first of the genotype, that is IAIB, we will get IAIO and IBIO. That means the phenotype of that F2 generation will be A and B. Similarly, in second case, when we cross this individuals with O blood group with individual his cousin with blood group A, who is having genotypes IAIO, then the F2 generation will be having IAIO and IOIO. That will be same. Okay, that means the F2 generation will having A blood group or O blood group. Same is with B. When B blood group individual is crossed with the O blood group individual, the cousin mating, in the F2 generation we will get one offspring having B blood group while other is having O blood group. So these are all the possibilities of the mating of among the cousins. So that will be the solution for the problem. The next problem is what kind of offspring would be produced in each of the following rabbit crosses. Now you just imagine, this is simply the process have been given. Capital C when crossed with capital C, small c. So it will give the same genotypes in F1 also. That is capital C and capital C small c. Similarly, in same second case, if this Himalaya, that is CHC, is crossed with another CHC, we will get first individual as a chinchilla, that is C having CH. Second individual having CHC, which is Himalaya. Sorry, in first case also we will get CH, which will be Himalaya, and in second case we will get CH small c, which will be also Himalaya. So all the individuals in the next generation will be of Himalaya. In the same way, you see the third one. In this case, we will get full color and Himalaya type of individual. So by seeing the genotype itself, by crossing, you can get the next generation. Similarly, you can assess the parental generation. So any of the type of questions you may get in the examination. So from this genotype, you can get the idea about the genotype of the offspring also and the parents also. So you take it as an assignment simply, and this is another example. Just these are for your practice purpose. The individual A is crossed with another individual. Now here the individual A might have the genotype of IAIA also and IAIO also, or IA small I also. So you consider all the possibilities and you try to make the matings among this individual and what will be the progeny or offspring of this individual that you have to find out. Okay. So this is the assignment for you. So that was the part regarding the multiple alleles. Now, second half of this today's practical is the sex-linked inheritance. So here also, before directly discussing the examples, we will see about what is the sex-linked inheritance. So as the name indicates sex-linked, it means these are the conditions where the genes are linked with the sex chromosome of that particular individual. Now we know that in the all the individual, the last pair of the genes is having the pair of the sex chromosomes. Now the genes which are located on these sex chromosomes, they are following some typical type of pattern for their inheritance, and based on this, these examples are there. So the genes which are present on the X chromosome, they are called as sex-linked gene, while the genes which are present on the Y chromosome, they are called as holocentric genes. Now, the same thing is, now we will discuss in detail. Here I have shown the chromosomes, that is X chromosome and Y chromosome. As we know that X chromosome is always larger than the Y chromosome. In this case, there are some of the region of this X and Y chromosome, they share the common region, where the genes which are located on this X and Y chromosome, they are complementary to each other, and that portion is called as homologous chromosome. So the homologous chromosome of this homologous portion, so the homologous portion of this chromosome is denoted here in this figure at the lower bottom. Lower part of this chromosome, and upper part is the non-homologous part, where the genes, they are unique genes, means the genes which are present on the X chromosome, they are not complementary with the genes on the Y chromosome. Here, as the Y chromosome is short, they carry the less number of genes as compared to X chromosome. Now, whatever the genes they are present on the non-homologous part of this X chromosomes, they are called as sex-linked genes. To be more precise, this is the most accurate definition, that the genes which are present on the non-homologous part of the X chromosome, those genes are called as X-linked genes or sex-linked genes. Whereas the genes which are present on the non-homologous part of the Y chromosomes, they are called as holocentric genes. So here, the genes which are present on this non-homologous part of the X chromosome, they follow crisscross pattern, or we also call it as a skip a generation pattern. Now, why it is called as crisscross pattern? It is called as crisscross pattern because these characters get transmitted from grandfather to grandson through the carrier method. That means the if the abnormal condition is there with the grandfather, it will not express in the F1 generation, because there will be another X chromosome to protect that particular copy of X chromosome, due to which it will be in a hidden condition in F1. But it will again express in the F2 generation. And the thing is that in F2 generation only, also only the males will be affected and not the females, because the female will carry 2 X chromosomes. And as the character is sex-linked character, it will be present always on the sex chromosome. And in the female, there are 2 X chromosomes. So the another normal X chromosome will not allow that. It will suppress the effect of that abnormal trait. But in case of male, there is only 1 X chromosome. And if that X chromosome, if the male will get that faulty copy of X chromosome, definitely that male will be exposed to that condition. Okay. So crisscross pattern or skip a generation pattern means when the character get transmitted from grandfather to grandson through carrier daughter, then that condition is called as crisscross pattern or a skip a generation pattern. Now we will discuss some of the examples based on this sex-linked inheritance. The first example is in Drosophila, the red eye, which is denoted by capital W, is dominant over the white eye, which is denoted by small w. If a white eye female is crossed with red-eyed male and also the F1 female with her father, what will be the phenotype of the progeny? Okay. So here also there are 2 parts in this particular example. So first we will discuss the first part, that is if a white-eyed female is crossed with the red-eyed male. So here I have shown the cross. When we talk about the red-eyed male, so definitely this W means it will be this W is a character which is present on the X chromosome, which is responsible for eye color. Now, as the male carry only one copy of X chromosome, I have shown it with capital W, and this Y indicates the absence of X chromosome. That means either we can show it with 0 or Y. Okay, but the female is white-eyed, so it will carry the recessive copy of X chromosome, and both will be WW, small w, small w. Okay. Now, after crossing these two parents with each other, we will get this male will produce the two types of gamete, capital W, and one gamete will be having the Y chromosome, while the female will produce both the gametes carrying small W. That is the copy of this gene with white eye. Then in F1 generation, we will get capital W, small w, and small w, small y. So here the female will be red-eyed, while the male will be white-eyed. Now, second part of this question is, when the F1 female is crossed with her father. Now here F1 female is having the genotype capital W, small w. When it will be crossed with its father. Now father is having capital W, Y. So this condition is shown here in the next cross. Here the genotypes of this female F1 female will be capital W, small w, and her sire will be having capital W, Y. So this F1 female will produce the two types of gametes, that is carrying capital W and small w, while her sire will produce capital W and Y type of gamete. And in the progeny, we will get capital W, capital W, capital W, small w. These will be the genotypes for the female, and for male, we will get capital W, capital Y, and small w, small y. So in both of the case, 50% of the all these females will be having red-eyed, but in case of male, 50% male will be having red-eyed, and while another 50% will be having white-eyed. So here, if we see in the beginning, the male will be having red-eyed, okay? But it in the F1 here, the males are white-eyed, because here it is not, this character of red-eyed is not expressed, because the gene responsible here is not expressed in F1, but it has been expressed in F2 generation. Okay, so this type of inheritance is called as criss-cross pattern. So the same thing is given here, that the characters has been reappeared in the F2 generation, and this type of phenomena is called as criss-cross inheritance. Then the next example is, a woman with normal vision marries with a man with normal vision, and they have a color-blind son. So this is some of different example. Here both the parent, they are having normal vision, but their son is color-blind. And then second part of this example is, her husband dies and she marries a color-blind man. Show the type and proportion of children those might be expected from this marriage. Now in the solution, we will see that as it is given that both the parents are having normal phenotypes, but their son is colorblind. That means one of the parent, and most probably it should be female only, should carry at least one copy of gene carrying which is responsible for colorblind, because the son is colorblind. Okay, and definitely if the mother is normal, that means the another copy of X chromosome will be the normal. So the mother will be heterozygous carrier. That will be that she will carry the XCX type of genotype, while the male will having the genotypes of capital XY. So this female will produce the gametes XCX, while male will produce the gametes X and Y. When they in the F2 generation, we will get XCX, XCY, XX, and XY. That means whatever the female progenies we got, among out of that 50% will be carrier and 50% will be normal, whereas among the male progeny, one male will be color blind, while other will be normal. Now we will see the second part of this example. That the husband of that female is died, and that same female has been married again, but this time she married with a color blind man. So this condition has been shown here. Now this female will have the genotypes XCX. When she marry with the colorblind man, the colorblind man will have the genotypes XCY. So from this, in the next generation, we will get XC, XC, XCY, XCX, and XY. That means the normal, the male we will get normal, and one female will be with color blind, because she will having the genotype XCXC, and out of the two male, one will be color blind and one will be normal, while one female will be also color blind and another will be normal but carrier. The next example is the colorblind man marries with a woman of normal vision, and all of whom were normal vision. Thus, the colorblind among the grandchildren are expected to occur if there are cousin marriages among these children. Okay. So here, this is the solution. The genotype of the male will be XCY, because the male is colorblind. Okay, here it is given that the man is colorblind and woman is normal, so she will carry capital X, capital X. So in F1 generation, we will get the females with genotypes XCX. So all will be normal but carrier, and the males will be normal. That will be absolutely normal, having genotype XY. Okay. If we made the if we make the cross between the cousins, it will be XCX and XY. So this XCX female will produce two types of gametes. That is, one will be a gamete which will carry the copy of colorblind, that is XC, while other will be normal with X chromosome. Then the male will produce X and Y gametes. Okay. So in F2 generation, we will get XCX, XCY, XX, and XY. So the grandchildren out of two male and two female, one male will be colorblind and other will be normal. Whereas both the females will be normal, but half will be homozygous, while half will be the carrier. So in this way, we have to conclude for this particular example. So in this way, in this multiple allele and sex-linked inheritance, mostly the examples will be logical. That means you have to, once you will know the concept of the multiple alleles and sex-linked inheritance, then before solving the example itself, you can have some of the idea regarding the solution of that problem. So I am very much thankful for all of you for paying attention to this lecture. Thank you.